We have a discussion session now, and we're going to be discussing two subjects that are mildly controversial in the field. And the first is about microglial nomenclature and states. And the second is about whether neuroinflammation is good or bad. So we're going to start with the first topic, which is microglial nomenclature and states. So you may be aware that there was a kind of position paper that was headed by Rosa Paoluccelli and Amanda Sierra and Yves Tremblay and Beth Stevens about microglial states and nomenclature and the kind of the situation that the field has got itself into, but I think most people would agree that although that paper well described the problem, it didn't come up with really good answers in terms of what we should be doing. So what we're discussing here is, for example, whether we so the field's gone through various ideas about microglial activation or resting states, about M1 and M2 states, and then states such as DAM or ARM or BAM or WAM or whatever you like, and whether there really are any states to microglia. So this is what was kind of partly set out in this position paper, but as I said, there's no clear answers. So we wanted to get some answers from our panel here. And Jenny, would you like to start things off?
Sure.
But you might then move on to Bart, so you might have...
Okay. I just found out I was doing this, and I said to him, I think this is a lot of navel gazing. So my view is, I think as Guy said, you know, we go through these fashionable phases of calling microglia DAM or M1 and M2, and next year it will be something else. And I just think, is it helpful? You know, is it more important to know what they're actually doing? And is it helpful to have a state? Because if you've got a state around a plaque, well, okay, you've got a state around a plaque, but then you've got another state somewhere else. Is that going to be helpful? If you're trying to develop a drug target, is it helpful to have those two states? I don't know what the answer is, but I do think that it's very difficult. When you get, I mean, today and yesterday we had a lot of these maps showing all these different states of microglia derived from, RNA-seq or whatever, bulk RNA or whatever we like to call it. And I do wonder, first of all, you're getting all this stuff. You're taking it out of a brain. How much of that is affected by you taking that out of the brain? How much is artifact? How much is really real? And I just wonder about that, and whether we're just getting bogged down by all these different ways of describing microglia when really we should be concentrating on what they're actually doing and how they're involved in disease. I just don't think it's helpful. So that's my view. I shall pass it down. Oh, we don't. Oh, yeah, no, we don't.
So you want Bart to speak next, is that correct? Because I could argue with Jenny.
Yeah.
For example.
Go on then.
Okay, so I do agree that this the way we kind of are emphasising named microglia now is all derived from RNA-seq, different types, different areas of the brain, etc., etc. And I do agree with Jenny on that it isn't particularly helpful, because for a start, we have absolutely no idea how that genetic information translates into proteomics and how that in turn translates into function, which is what we should be all after. So there, I agree completely. But I do actually think it would be really helpful if we could get some sense of a nomenclature that means every paper is going to add to the literature because we know precisely what we're talking about. And we don't have these invented new terms that just complicate the issue. So it would be really nice if we had something, some sort of a baseline, whereby each contribution from every author is going to be significant, it's going to be important, and it's going to build up a story. So that's, for me, the advantage of having some sort of nomenclature. Now, I think that it would be really foolish of us if we didn't agree that there's some sort of a homeostatic state, not necessarily defined by a collection of genes, but by the function. The cells are able to function properly. What do? They're able to move, they're able to phagocytose, they're able to respond to stimuli, etc. I think if we could always marry a function with a marker or a function with a set of genes, then we'd be getting somewhere. So the argument is that I think there might be a use for nomenclature if we were able to ensure that it is added to the body of work that's out there, not confuse the issue, but instead illuminate and always link a function. Next.
next in line. I'll try to give an answer and try to ask a question. So the answer to some of the questions, how useful is that or was that? So I remember the times we used to stain with tomato D-lectin and maybe even more weird markers, and we got a very bad grip on what we are seeing. And in that sense, it was good that most of the editors were falling in love with the Disney plots, and we all had to do the transcriptomics virtually in every paper now to get it published. But the big but is in my view, we're still 2 stages away. We miss a lot of proteomic data, we miss information, and we also know that proteomics as it stands right now is limited as a technique. We could use ribotech approaches, we could combine these approaches to get more insight on which of this protein, which of this transcriptome is being translated into a meaningful protein. And I had this discussion not too long ago with Ido Amit, and because we had data in my lab where a whole transcriptome was only translated by about 5%, and in his hands it was 7%. So which means, in other words, if 95% is not being translated, doesn't mean it's not important, but maybe not important for function. And so I think we miss a lot of information. The protein level is not the ultimate level we need to go. I think we all know that there are a variety of really important post-translational modifications of proteins which actually determine what these proteins are doing in a biological system. So we need to go to the proteome level and beyond to look at the post-translational modifications in much, much greater detail. So I believe, in summary, the transcriptomics have been helpful to show us that there are various differences between cells. I still find for myself we haven't really made a lot of effort to find out the dynamics of these changes, because most of the transcriptomic studies are of cross-sectional and not of longitudinal nature. So we have a certain, like a snapshot of what happens in the cell, but we do not know where does this cell transcriptomically come from and where does it go to. And so this is something which is still missing and could probably be done by transcriptomics. I don't think it's completely useless, but I think we need to get to the next stage.
Yeah, so maybe I was in the discussions with this position paper, and the guy commented there's no real guidelines, but let me, a little inside look, right? So we're all bred to be opinionated and stubborn as scientists, and then try to get 100 people in one Zoom to converge on a nomenclature. So if you raise your hand if you think that is a probable cause. Okay, that's solved. So that's the reason that doesn't happen, because there's no way, because everybody coins his or her own favorite label, because it's branding, in my opinion. So it's branding. So that said, I find it hard to believe that you find transcriptional diverse cells that has no biological relevance. I find that difficult to understand. I find that very difficult to assume. So why would nature go about creating these cells that all have different RNA contents. Do we measure everything? No. Do we know what they mean? No. But I find it difficult to imagine that none of these different states that we map are biologically inert. I find it very difficult to imagine. The reason I think we find so many states in transcriptomics is that the technology is more advanced than all the other ones. It's just you get high-dimensional data, so it's much easier to identify different states, and then I think time will tell how many states we actually have, and there's several groups in the world that are embarking on this to really figure out if these states translate into functional states. But it's not so easy, because as Michael already said, we do a lot of cross-sectional data. Mimicking human microglia in a dish is extremely difficult. To reconstruct these states is extremely difficult. Mice are, well, I would argue that mice are not humans. Maybe you disagree, but I think that mice are not humans. So we cannot model every human state in a mouse. So I think it's just, it's complicated. And I think but I strongly agree with the notion in the paper that suggesting that these cells have, let's say, M1, M0 binary states is a gross oversimplification of reality. And I would strongly argue against using that terminology. I think Richard Ranselhoff I hoped he effectively buried it a few years ago, but apparently not. But I think it's a very, this is discussion, right, so I can be rude. I think it's an absolute oversimplification of reality if you stick to M0 and M1 classifications. And if you disagree,
And M2.
let's go.
Yeah.
Yeah. So maybe my perspective comes a little bit from the neuroscience background which I have developed. During my PhD, I've been working in the retina. The retina is a beautiful structure. I've showed you in my presentation the picture. It's very layered. It's one of the best described morphologically brain regions. So we know, but even so, we morphologically have it described as like 60 different cell types, but it could be then also 80 cell types. So there is some discrepancies on some of the points. So I always think like that the morphology and how these cells get input and output is a critical parameter. That's why I put a lot of effort on it in getting to know how the morphology could give us some kind of ground truth on which we can then build the transcriptome on it. So what we did, for example, at that time, we morphological had distinct cell types. We had the photoreceptors. We have certain types of amacrine cells, horizontal cells. Also microglia were there, and we just transcriptionally profiled them and looked what are transcriptional difference. And we found for each of these cell types a set of specific transcription factors, a set of specific disease-associated genes. But at the same time, we also know that there are certain complexity in there. For example, there are this starburst direction selective amacrine cells, which respond when the light comes from left to right or not from right to left. And we know they have the same transcriptional signature, they have the same morphology, but then the distribution of the receptors within the cell is different. So I think there is a multi-layered structure of complexity, which I have learned from the retina, that I also anticipate we will experience in the microglia field. And we should be open to this perspective and not just like stick on one particular paradigm. I'm also thinking there's also a lot of like, we found a new marker, TMA 119, that's ground truth, it's labeling exclusively parenchymal microglia in mouse and human. No, it does not. And I have a really hard time to fight this in the retina. For example, it has been shown when you do optic nerve crush, no, not microglia, there are other cells labeling. Or when I differentiate retinal organoid, generate retinal organoids, and we have microglia in there, and we label TMA 119, use TMA 119 staining, we see osteoblast labeling, because early during the development, also osteoblast expressed TMA 119. One reviewer sticked on it, we should show that this TMA 119 is expressed in the IBA-positive cells. What can I do? And I think this is like the point where I'm like, you can get easily frustrated when you are like confronted with this hardcore truth and the people are not looking at the data. And we tested several different antibodies and it is just how it is. And we cannot change this. And I think this is something that we should be open about it, coming from this perspective.
I think because the time is short, we should open it up to the audience, whether they have anything they want to share with us about nomenclature or states, or any question they want to put to the panel.
Coming back to the transcriptomics and sort of all omics, really, I think one of the huge issues is actually accessibility of that data. So I think some sort of alumni from the BARS lab are really leading here, especially sort of Shane Lidlaw, in creating websites, searchable tools that you can use to research different genes and things like this, so that you don't have to have specialist training in bioinformatics to actually get the information that you need from these data sets that, rightly so, are made open access. But without that training, sometimes it's difficult to find out what you really want to find out, and it might not be written in the paper, or it might not be listed in the paper. So I'd really like to see those sort of tools used a lot more.
I think there's also another factor that you should consider, especially with transcriptome data. It's very difficult to make a metadata analysis of all the transcriptional data, putting it together. So like ideally we have like transcriptome data from different groups, but as soon as you would like to merge the data together, it's not possible, or it's very tricky because you have to control for like algorithm that, to make sure that the difference you're seeing is truly biological or not just an artifact due to the methodology or other factors that are in there. And I would love if we could have a huge database with all the microglia, the transcriptome data coming together to get an idea of the transcriptional signature. So I would be totally on board on this.
Yeah, we tried a few years ago, and it's actually very complex. So we built a database, started to download all the data, uniform it, make it accessible, but you basically need a truckload of money. That's the problem, because you need one person to full-time do this and to keep the website updated, operational, and you need a lot of bandwidth if people... So one simple paper is easy, right? I mean, one paper, one data set, you can make it a shiny app and make it accessible. That's easy. But we tried to build a website, download... So first we had to curate the data, because I won't tell you how horrible some of the data sets are. So we had to curate a lot of data sets, and then we started to analyze them and to implement them in the web. It's just a ton of work. And
I think it's really worth it, because
that
one website, Brain RNA-seq, has been like invaluable, especially for our lab. It's been absolutely fantastic.
Yes, I agree. But
it might cost a lot, but I think it's definitely worth it.
I agree. As a community, it would be very valuable, but one way or another, we need to raise the funds to do it, because to do it on the fly, as I tried, it's just not sustainable.
Thank you.
I think we did something with Fargo, with all the... Remember, we looked at the comparison between different sites, and we did... who was it who did the RNA-seq? It was you guys, wasn't it? Life and Brain. And then we put that somewhere. It's got to be on the database still. I think it's kept live. Is that right, Angela? Do you know if it's still live, that database, when we had all the comparisons between all the different... We made different microglial preps in different places around Europe, and then they were all collated. I'll have to check whether it's still live, because that was the data set of all the different microglial preps. iPSC microglia.
Just a comment, really. To what extent do you think the different microglial states are predetermined versus stochastic, given the plastic nature of these cells?
Asking
anyone.
Oh.
I think they're so plastic, I think that's impossible to say. I think they're so influenced by what's around them. It would be difficult to say whether it's predetermined.
One level we rarely touch on is that, especially in chronic disease, we may have different generations of microglia cells, because there's proliferation and there's death. And a microglia cell which populates once a healthy brain is a thing that gets different cues and different pattern recognition receptor stimulation as a microglia cell that is being born into a diseased brain, into, for example, an AD brain. That's like a kid being born into a war zone. It gets very different cues. It will react differently throughout its life. So we know that we have these different generations there. We have dying, and we have death, and we have microglial proliferation. Still, we look at one transcriptome, so to say, but we do not know which part of that transcriptome comes from, so to say, F0, F1, F3, F4, and Fx generation. And I think this is a very complicated thing to answer, because, and it's likely that the different generations execute different roles, so to say, and different activities in the brain. I think this is an unsolved question.
Now, Bart mentioned the kind of finances, and Michael mentioned that it's more or less required now to do a transcriptomic analysis of your microglia in some disease state or physiological state. And that raises the issue of, I think a lot of us have encountered this. You may be doing a paper on, some physiological effect of some receptor, and a reviewer says, well, what's the transcriptomics? So do we feel as a field that it's essential as a way of characterizing microglia to do the transcriptomics? Because that excludes a lot of labs and people coming into the field. But I know that, but if it is required, then maybe it is essential. But transcriptomics is only one way of characterizing the phenotype of microglia, and there are a variety of others, as we've heard. But do we feel, as a field, that it's essential to classify your microglia into BAMs or DAMs or WAMs or whatever it is?
I mean, I think it's helpful if someone else is looking at another receptor and you've got your gene transcripts and you want to see if it's sort of feeding into the same transcriptome. I think it's helpful from that point of view, but I don't think it's essential. But, it depends on the reviewers, doesn't it? And maybe we need to be educating the reviewers that if you've got other evidence for how this receptor works, that is a function that's important. But for everyone's information, it's useful to see how it actually affects the transcriptome. But then, as I said earlier, it's really the function that should be most important.
I find it always a little bit disappointing because what always happens is always the same transcriptional genes are shown. Yeah, it's always the same. It's always like IBA1, P2Y12, and all these markers. There's nothing novel in there. And that is like, I feel like a waste of potential that we could have. I see more like it would be useful to look, for example, not just like at single cell microglia population, but looking in the entire brain region on how the different cell types interact with the neurons. So this is cell-cell interaction, I feel like, has a much better insight and could give us a better intuition, what are the microglia doing and what are the receptors, how they're connecting with myelin cells, with astrocytes, with neurons, which receptors there, what ligand receptor exchanges there. And I see that there is some movement in this direction, which I'm very much looking forward to see what will come up in this direction.
I think one of the issues about just demanding transcriptomics is that you're ignoring all the factors that interfere with a potential group of signals. So, for example, cell-cell interactions, it wouldn't be surprising at all, and I think Michael might be able to tell us about that, whether the transcriptomic situation is different if you have one of your microglia TNTs making contact with the neuron, for example. And equally, we have all these other factors that we know alter microglia, for example, development, adult, age, neurodegeneration disease. That sequence of events all have a very significant part to play. We have the influence of sex, we have the influence of a load of other factors. And I just think that demanding transcriptomics in always is foolish. It just doesn't get us anywhere. I
But yeah, I agree. But it's also a bit. It's not like reviewers as a different species. It's us,
Yeah.
right? So we are asking ourselves to do that.
I don't.
But it's not like reviewers are like sitting in the next room over and they don't know what we're doing, but they're just asked for omics. No, that's not. It's us.
So I think it really depends on the scientific question which is being addressed. I think there are questions. If the question is there a whole population change? And I like this term married to function very much because I think this is really the important part. Then it may make sense. But we also have to, I think Guy said it quite rightly, it keeps a lot of people and a lot of groups outside of the field, which we could need to advance as a community. And my concern is by now we have this request to do the transcriptomics. And then where's the end? The next thing is, I'm waiting to that. The next thing is to connect the transcriptomics to spatial transcriptomics. And after that to spatial proteomics. So it's like, it's an endless thing. And I think we, as authors or as when we design the studies, as well as reviewers or as editors, we have to really refocus what is the original scientific question we want to ask in this work, and then really tailor the methods to answer that question. And it could possibly involve transcriptomics at some stage, but not necessarily the every work, so to say. Because where is the end? Next level is we have to do spatial transcriptomics and something after.
Yeah, I'm wondering about alternatives. I'm also working on T cells and B cells, where you have a bunch of markers that very well describes a cell. So you can apply flow cytometry for following changes in cells. And I think a microglia, that is more complicated. How you see that? So is probably transcriptomics the best we have right now?
Well, I think one of the problems about flow cytometry is that we're relying on markers, and we already have heard that, for instance, the gold standard marker is not truly a marker of microglial cells. So there's an issue, I think, with flow cytometry, as there is with every technique. So there's there's no doubt there are merits in using flow cytometry, but we have to be aware of the issues that arise because of the fact that we don't have full details about how specific markers are.
Working.
Sorry, my bad. Maybe this is a bit off track, but also similar to what he asked. What is your opinion? I mean, we spoke a lot about primary microglia from human and mice and also iPSC ones, but what is your opinion about monocyte-derived microglia, which is being studied quite a lot these days? And again, it comes down to the point that if you want to differentiate macrophages from microglia, just the surface markers are not enough. So as experts from the field, what is your opinion about it?
Well, I suppose you've got to talk to Janu about that, because he thinks that microglia are MIB-independent and macrophages are MIB-dependent. So he would say that there's different populations, and so unless you're really sure how those populations are different, trying to say that a monocyte-derived macrophage is really a microglia is a bit tricky, isn't it? I mean, what else is different? We know that different markers on microglia are different from markers, we're back to that, are different from macrophages. So I would say it's tricky. I would say you'd have to be careful. Why,
I'd say you have to do transcriptomics.
yeah.
Spatial.
No.
Spatial.
Yeah.
Again, it may really depend on your question. And there might be some things you may be able to answer with a more simplified cell line or approach, and others are not. Jenny may not know that, but she published a trends in neuroscience paper in 2007, which really influenced my thinking, together with Helmut Kettenmann, showing that microglia carry virtually every receptor for neurotransmitters on their surface. And you bet these neurotransmitters very strongly influence how the
on that lot, shouldn't I?
how the microglia behave. We actually did. And you know, I'm interested in norepinephrine. Other people are interested in other neurotransmitters. But whatever you look at, they very much shape what the microglia do, what they express, and how they react and how they function. So in the brain, we have a very specialized situation. In each brain region, so to say, you have a slightly different profile of neurotransmitters because the neuronal population differs, right? So the influence on that particular subset of microglia cells in that region may be different because you have a difference in your neurotransmitter profile. And so it may take more than a macrophage to model what really is the case with microglia cells, because they have this influence.
Last question.
Hi, yeah, I just wanted to give an industry perspective. So I work at AstraZeneca. We're working on microglial targeting for drug discovery. Maybe we've moved away as well from obsessing about the transcriptome when we develop drugs that activate or block microglia. And we're now trying to focus on the proteome, the secretome, the metabolome, and then integrate all that data to understand how we're changing biology and function. It's a pragmatic approach as we understand the complexity of biology. But my main question to you is, do you think we need to be targeting different subtypes of microglia for drug discovery?
Yeah.
And how are you going to do that? Well, I mean, as Michael just said, you know, depending where you fish them out from the brain, they're going to be influenced by the environment they're in, they're going to be influenced by the neurotransmitters. So from that point of view, yes. It's just how are we going to do it? You know, it could do RNA scope on a section or something and say, well, yeah, or...
A simple example is if you're doing 2-photon laser scanning or 3-photon laser scanning microscopy of the neocortex and the hippocampus, both in an APP or any other mouse model, microglia cells have different motilities, different scan times, different branch orders, and so on and so forth. And I think we should probably, this is in a 3-dimensional context in a living mouse, which is non-anesthetized, walks a treadmill. And I think this is something we probably could use more in future. We've been looking to fix tissue for 600 years by now. And now since maybe a decade or so, we can look to the living mouse brain. And that probably helps us to get to the next stage. And I only can tell you from my own experiences, the microglia cells in different brain regions have really, even under homeostatic conditions, different qualities, quantities, and different properties. And I think we just can't ignore that.
Great. Now that we've solved the first question,